Hello everyone, welcome to Faster Classes. Today we are going to start the part 2 of our chapter 10, Microbes in Human Welfare. In the part 1, we have done household uses of microbes and industrial uses of microbes. In this part, we will study microbes in sewage treatment and microbes in biogas production. Let's start now. Sewage is large amount of wastewater generated in cities and towns. It is also known as municipal waste, and it consists of household waste, human waste, and laundry waste, etc. And before their disposal in natural water bodies, they need to be treated in sewage treatment plants. And this sewage contain large amount of heterotrophic bacteria, and these bacteria are going to help us in our sewage treatment plant. Sewage treatment is done in two stages: primary treatment and secondary treatment. Primary treatment involves only two major steps, that is filtration and sedimentation. Floating debris is removed by sequential filtration. The debris or the waste which is floating on this water, on this sewage, is removed by filtration, while the grit or waste that can settle down, like mud, soil, which settle down, is removed by sedimentation. The particles which settle down is known as primary sludge, while that lies above is known as primary effluent. This primary effluent is now released for secondary treatment in aeration tank. In this aeration tank, a constant supply of air is given, and this constant supply of air helps in the vigorous growth of useful aerobic microbes. Aerobic microbes are those which require oxygen. These microbes forms flocs. Let's see the definition of flocs. Flocs are masses of bacteria associated with fungal filaments to form mesh-like structures. And these microbes, they consume the major part of the organic matter in the effluent. See, this effluent contains a large amount of organic matter. For their consumption, we need aerobic bacteria. They feed on all these organic waste and reduces the BOD. What is BOD? BOD is the biochemical oxygen demand. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by the bacteria. See, in 1 liter of water, whatever amount of oxygen we require for the oxidation of organic matter by the bacteria is known as BOD. Greater the BOD, more is its polluting potential. See, if the BOD of wastewater is more, it means that more bacteria are going to utilize more oxygen for the oxidation of organic matter, and hence we can say greater the BOD, greater is the polluting potential, or greater is the organic matter. When BOD of sewage decreases, effluent is passed into settling tank where bacterial flocs sediment. After BOD test, when the rate of BOD decreases, and that effluent is now passed in the settling tank where the bacterial flocs which were formed in the aeration tank, they settles down. And this sediment is known as activated sludge. The bacterial flocs with sediment in the settling tank is known as activated sludge. Some amount of this activated sludge is pumped back into the aeration tank to serve as inoculum. Some amount will help as a starter for the next effluent which is going to come in the aeration tank. Major part of sludge is pumped into large tank called anaerobic sludge digester. The rest part of this activated sludge is passed into anaerobic sludge digester where anaerobic bacteria, they feed on them and lead to the production of methane, hydrogen sulfide, and CO2 gases. These gases together can be used as biogas and can be used as fuel, while the effluent from the secondary treatment plant is released into natural water bodies. Now the effluent, or we can say sewage, which is less harmful now, is released into natural water bodies. Now we will see microbes in production of biogas. Biogas is a mixture of gases containing predominantly methane produced by the microbial activity and used as fuel. The biogas is generally where the amount of methane is generally 50 to 75%. 50 to 75 methane is present, while we can say 25 to 40% we have CO2, and the rest like H2S, hydrogen, ammonia, these are present in trace amount. And there are certain bacteria which grow on cellulosic material and produce large amount of methane, CO2 and H2. This group of bacteria are known as Methanogens. And the bacteria which is responsible for the generation of these gases is known as Methanobacterium. And these bacteria are also present in the rumen of cattle that help in breakdown of cellulose. Rumen, it is a part of stomach of the cattle or ruminants. That's why humans are unable to digest cellulose, while the ruminants or cattle, they are able to digest cellulose due to the presence of this methanobacterium in their stomach. Now let's see, excreta of cattle vobber is rich in these bacteria. Vobber of this cattle also have this bacteria, and this can be used for generation of biogas or vobber gas, and this vobber or excreta of cattle can be used for biogas production. Now let us see a biogas plant. First we will see the constituents of it. A concrete tank which is 10 to 15 feet deep, in which bio-waste are collected and slurry of dung is fed. Then we have a floating cover which is placed over slurry, which keeps on rising as the gas is produced. We have two outlets. Outlet 1 connected to a pipe to supply biogas to the nearby houses, and outlet 2, spent slurry is removed through it and can be used as fertilizer. See, from here, dung, household waste, it is fed into this large tank, and here the bacteria which are present, we can say methanobacterium, which are present, they lead to the production of gases like methane and CO2. And as the level of the gases rises in it, the floating cover which is present, it also moves up. Through this, the gas moves through the pipe to the nearby houses, and the slurry which is utilized, which is oxidized, is removed through this outlet in another chamber, and it is used as fertilizer or manure. Biogas produced is used for cooking and lighting, and this biogas is predominantly used in the rural areas of our country. There are two associations or institutions which are responsible for developing this biogas technology in our country. They are IARI, Indian Agriculture Research Institute, and KVIC, Khadi and Village Industries Commission. These two institutions have led to the development of this biogas plant. And today we are done with the two topics, microbes in sewage treatment and microbes in biogas production. In the next video, we will start with microbes as biocontrol agents and microbes as biofertilizers, and that would be an end of this chapter. And you can download the notes, the similar notes, from the description box. Till then, thank you so much.